Okay, good to go here. So, thanks everybody for coming yet again. We got, I think, three more weeks of the seminar. So this week, myself and Dr. Oliger here talking about the applications. Next week we'll discuss analysis and visualization methods. See me again and Jason Crane as well. And then in the final week, you're going to hear about overview of applications for cancer and neurological diseases. And then that's it. And then the plan for today is there's going to be a hands-on lab at the Surbeck lab, so it's the human 3T system there. I heard they were trying to get a rat on the table over there to actually do a sort of a full mock study there, and so hope you guys can check that out. And I don't know if we talked about this, but the other thing, we've been posting these lectures, the recordings of the lectures on YouTube. I don't know if the playlist has been distributed widely, but we do have those, so you can come back and look at those later on, and we have the slides available too. So, YouTube, like anybody YouTube. So, not like who's gonna see. Yeah, that's right. You're gonna get tagged, filtered from the other 10 views or something. And so the other thing is, if other people that you're working with, new people that come into your labs, hopefully, because we offer this every 2 years typically now, if they come in the meantime and want to catch up on some of this, make sure to have that for people to point to there. So I'm going to spend the first half-ish of this talking about giving sort of a pretty basic overview of the application of hyperpolarized C13 to the heart. And do my best to get everything correct here. This is kind of a new sort of research area for me, and so something I'm learning with you guys. So I'll talk about really probably the most important part of this is talking about the cardiac metabolism and energetics. So what do we expect to be able to measure by hyperpolarized C13? Then what is known about how the metabolism is going to vary in heart diseases, or even there's quite a bit of normal variation in cardiac metabolism as well. Point out a few interesting things that I came across for some other agents beyond Pyruvate, and talk briefly about the imaging methods. So the heart is extremely energy hungry, as you might imagine, producing a huge amount of ATP every day. So obviously it needs a lot of sources of energy to create all that ATP. And the heart is relatively flexible, or at least the healthy heart. It's able to use a number of different energy sources. What I've seen is that it's the fatty acids are the main source of fuel for the heart, but also the heart can do a lot of carbohydrate metabolism, so glucose, taking up glucose or lactate from the blood, converting on this chart on the right here. That would be going into pyruvate right there. So pyruvate coming from either lactate or glucose, other sources. The heart can also use ketone bodies and amino acids. Sorry, this should say branch chain amino acids, I believe. This is some specific work looking at these, but And then one of the things that I've heard talked about a lot when talking about heart metabolism is this thing called the Randall cycle. And it's described as this reciprocal regulation between this fatty acid and glucose oxidation. And that is what enables the heart to be, so the Randall cycle is what enables the heart to take advantage of basically the energy energy sources, I should say, that are available. So this is, good for you. I think it makes it very, and this is unlike the brain also, that we'll hear more about, which is very much glucose driven for its energy. The heart is more of an omnivore. It's going to take up whatever various sources of energy is shown here. This, I think, is a big challenge for cardiac metabolism studies in general, is that how your heart is functioning is going to be very sensitive to your circulating levels of blood glucose or fatty acids. So that's largely driven by your fed or your fasted state. And I'll come back to that a little later. And then there's a lot of interest in doing hyperpolarized C13 for cardiac disease, because there's a bunch of places where there's a lot of evidence that there's either metabolism is either a cause or an effect of heart disease, and cardiovascular disease just being such a huge source of death in general. And a lot of the hypotheses that I've seen is that it's really this some of the metabolic changes that we can hopefully measure, and that are just driving the disease, is this issue of substrate selection. So by that I mean, how is the heart preferring to use fatty acids versus perform carbohydrate metabolism, versus ketone bodies as its energy source, and sort of dysregulation, probably with maybe with the Randall cycle, or and the heart showing these shifts. So like the obese and the diabetic heart, the data shows that there's an increased reliance on fatty acids for this oxidative energy production. Whereas sort of the opposite effect, at least in terms of the fatty acids and carbohydrates, is that there's a shift from the fatty acid oxidation to glycolysis, so basically the carbohydrate metabolism, and heart failure and some hypertrophies. This statement here, there's a bunch of different sources of hypertrophy, and so this could actually vary between different forms of hypertrophy, which is basically enlarged heart muscle. So larger muscle, and it can come from diabetes, it can come from genetic disorders, and it can even from intense exercise. You can thicken your heart. Then another thing that's been studied a lot in the hyperpolarized field is looking at ischemic heart disease. So this would be analogous to like having a heart attack, a blood clot into coronary arteries of your heart that prevents perfusion to part of your heart muscle for a little while. And we'll show some evidence later. You see markedly increased lactate, decreased bicarbonate production at the site of the injury. So here's just a little bit more detailed view of the metabolism and cardiomyocytes. So these are the main cell type in the heart that are performing the contraction to pump blood through your body. And this point out this review here is I'm very thankful for this review. It's cardiac. It's all about hyperpolarized carbon-13 for cardiac applications. So this is a really great resource. Very thankful to Damian Tyler and his group for putting that together. So here you have in the cardiomyocyte, this is being your sort of carbohydrate or glycolytic metabolism pathway here. So we see our usual glucose becoming pyruvate, and then the sort of mainly the usual story here. Pyruvate coming, and you see high amounts of PDH flux when the heart is doing a lot of carbohydrate metabolism. And then this other side here is the FFA here, standing for free fatty acids. So this is the fatty acid pathway. And both of them end up, the sort of question here is which one of these pathways is being used to create acetyl-CoA to go into the TCA or the Krebs cycle here. So this acetyl-CoA can be derived from fatty acids through beta oxidation, or it can be derived through the glycolytic metabolism pathway as well. And then I guess you can get acetyl-CoA from ketone bodies as well as these branched chain amino acids. So here's what some typical spectra in the heart look like following pyruvate injection. So on the left, you have a C2 pyruvate injection. On the right, you have a C1 pyruvate injection. And then this, the green dots here representing where you can follow the C1 label on pyruvate, and the blue dots representing where you can follow the C2 label. So the heart, you'll typically see a lot more, potentially more bicarbonate than you might see in other organs, depending on the fed-fasted state. And that's a result of seeing C1 pyruvate go through this, go into the mitochondria, experience PDH flux, and so then have carbon dioxide and bicarbonate created as a result of really the carbohydrate or glucose metabolism. But if pyruvate is not being used as a energy glycolysis is not being performed, then you would have pyruvate more likely probably going into lactate or alanine as opposed to going into the TCA cycle. And then because you can have pretty high levels of glycolytic metabolism, you will see a number of TCA cycle intermediates. So comes into acetyl-CoA, you'll see this exchange with acetylcarnitine right here, which would sit sort of somewhere in the middle of the 170-ish ppm, mid 170s ppm range. Then coming through the TCA cycle, first see citrate, appearance of citrate, and then if this label continues on, it'll reach alpha-ketoglutarate in exchange with glutamate, and then typically you'll see some both citrate and glutamate in these spectra. And then with the C2 pyruvate, you can also observe lactate and alanine. These are doublets because there's a directly coupled proton to the carbon in the 2 position, and so also consequently these have very short, relatively short T1s. So for observing lactate and alanine, it's much more preferable to be looking at the 1 position. C2 pyruvate, as you may remember, you're going to be able to see these TCA cycle intermediates. So a big effect in the heart is your fed versus your fasted state. There's substantial changes in cardiac metabolism between fed and fasted states. And actually for other organs, you should be thinking about this as a possible confounder of your experiments as well, because this can also affect metabolism in other organs. I think there's even a paper evidencing that some cancers will change in fed versus fasted states. And the difference is that when you're in the fed state, you typically have much higher blood glucose levels. versus in your fasted state, you haven't eaten in a while. There's much more free fatty acids in your blood. So you can see this in all of these results here. In the fed state, since you are having increased PDH flux, you'll see increased bicarbonate from C1 pyruvate. And that's what the spectra is showing, is increased bicarbonate in the fed state, as well as this nice image in a pig model here of the heart. And so, you know, this is potentially can have a big effect, right? This is pretty substantial, from almost no observable bicarbonate to quite a bit in both of these cases. So the experimental protocol that many sites have done is something like this, where you would fast your animal or your human subject, maybe overnight or for several hours, and then give them an oral glucose 1 hour before hyperpolarized study. I don't know what that is. It's special. No. So oral glucose 1 hour before the study. And the idea there is, well, on one hand, bump up the metabolite signal. You can actually see you see a lot more metabolites from pyruvate when you have a greater availability of blood glucose, and so your heart is performing more carbohydrate metabolism. And then the other point there would be, you want to have some sort of standardization. You don't want your subject coming in and you don't really know where they're at in terms of their blood glucose or free fatty acid levels, and so you could add a huge amount of variance to your data without knowing that. This is an interesting recent paper that did a bunch of really deep from Aarhus, doing a bunch of detailed pig studies. And they suggested, well, maybe based on their results, you could, instead of doing this oral glucose, you could just provide access to carbohydrate-rich food before the examination as an alternative. So get somebody in the fed state, not with the oral glucose, but carbohydrate-rich food, and to increase your metabolite signal, and without adding much variance. So it's I think this is a major challenge here, though. And then on to some of the applications here. So I mentioned before this ischemia injury. This is where you take the coronary arteries, come down the sides of the heart here, basically block one of those off. That restricts or maybe eliminates the blood flow to that part of the heart tissue. You can see this is where it's created this injury. And in that state, after this occlusion, you see an increase in lactate, a decrease in bicarbonate as the tissue is becoming not viable. And they're looking at the lactate to bicarbonate ratio, which is used by a number of studies, an interesting marker of the switch between TCA versus metabolism versus lactate metabolism. And then here they observed that even though you had this persistent defect here, I think on this LG's late gadolinium enhancement, the metabolite, the metabolic state actually returned to the baseline. There's a lot of interesting work in this area. Then this is just one example of a dilated cardiomyopathy, where they pace the heart, so make it go faster. So then it starts getting bigger. It's faster, bigger to compensate. And then this was a C2 pyruvate study, and they observed that this reduced the glutamate from C2 pyruvate. So that means instead of pyruvate coming down in that previous, or here we got it here, maybe not. So pyruvate coming from, into becoming acetyl-CoA, and then the TCA cycle, maybe this is increased reliance on the fatty acid metabolism, and seeing these changes in sort of an early state before the actually thickened heart, the cardiomyopathy here is developed. And then there's been one publication in the peer-reviewed literature. These images are kind of famous now. I've seen them many times from Toronto group of their first 4 human volunteers. And here they're following that protocol that I showed earlier of giving an oral glucose load 1 hour prior to the hyperpolarized injection, and able to observe pretty nice bicarbonate, lactate, and pyruvate here. And you see this distribution of bicarbonate being produced within the muscle, so myocardium, from carbohydrate metabolism. You see lactate everywhere. This is consistent with our observations as well. So this suggests you have actually a bunch of lactate in the blood pool as well as, I think, in the myocardium. And then finally, pyruvate is mostly in the blood pool here as well. And since there's so much signal here, you know, the resolution is actually pretty decent for these. I should say, I don't think I put it in here, but there's some they did these with surface coils placed on the heart, the chest wall. Okay, go through just a few other agents kind of quickly here, but one of the other things that's really interesting potentially with hyperpolarized C13 is trying to, instead of saying indirectly at the fatty acid oxidation, so inferring from less say PDH flux, that there's increased fatty acid oxidation, to actually try probes that are, you can do polarized short chain fatty acids like acetate. And in acetate, you see, I can see acetylcarnitine, and you have sort of reasonable polarizations and T1s, a little bit on the short side, but you, as you may recall from some of the other lectures, larger molecules are harder to polarize, so polarizing larger fatty acids is quite difficult. Another interesting agent along that line is butyrate, which is also a short chain fatty acid, similar T1 and polarization. And here you're, and actually is taken up by MCTs, the monocarboxylate transporters, which are the same transporters that take up pyruvate and lactate. So that's one of the parts we need for the experiment, right, is the agent to be rapidly taken up. So I think that's kind of promising there, and you can see a number of sort of some of the TCA cycle metabolites, as well as the acetylacetate and acetylcarnitine before you actually get into the TCA cycle. And then I thought this was a pretty cool study, actually co-polarizing C1 pyruvate and C1 butyrate. Apologize, it's kind of small here on the bottom, but one of the big questions in the cardiac metabolism is this: which substrate is the heart going to choose? Is it going to choose carbohydrate metabolism, going to choose fatty acids? And so in this case, you can actually create this condition within your hyperpolarized experiment. You inject both of these compounds together, and there's just you inherently have this substrate competition for which one's going to get chosen. And by choosing, I guess for you have to be, if you did C2 pyruvate, it would actually be bad, because then you'd get citrate from pyruvate. So C1 pyruvate in this case is better, and C1 butyrate. And also if you use acetate here, there's some spectral overlap as well. So this is pretty, and the conclusion of this paper was they could actually observe different results between injecting one compound at a time versus when they're injected in competition. There's been some work using Urea, looking at perfusion. These are rat heart images, so that's pretty impressive. There's been some pH imaging work couple of the things that I had seen were using pyruvate for intracellular pH. So there you're looking at pyruvate that's being taken up into the cell. That's being the label is going into bicarbonate, and then you're measuring the bicarbonate CO2 ratio, which is only happening intracellularly, as opposed to if you directly inject bicarbonate. There can exist both in the extracellular space as well as possibly in the intracellular space. And you can actually image, CO2 because there's quite a bit of, pyruvate uptake and quite a bit of metabolism in the heart. And then lastly here, the imaging methods. As you can imagine, there's extra challenges going to the heart because primarily of motion. There's the fed fasted, which is the physiologic challenge. There's this, and then motion challenge. Also, I think resolution, is tricky. You saw those images. There's a bunch of metabolite in the blood and in the myocardium. We're probably not so interested in what's going on in the blood, but very interested in what's going on in the myocardium. So for motion, you use triggering the cardiac or respiratory cycles and breath holds maybe. Some of the best images you see use some variation of a sequence like this, which is a spectral spatial excitation and a fast spiral readout. Spirals are really the readout of choice for fast cardiac imaging. And even that acetate result I zoomed through a couple slides ago is this ideal spiral CSI plus a spectral spatial pulse here. So again, spirals for their motion properties and spectral spatial to enable fast imaging. So you can do one TR, get one image, no chance of blurring across multiple TRs. And that's probably the least I've ever talked about in a presentation about whole sequences. So are there any questions? Okay, thanks. So I'm gonna turn it over to